thanks for the invitation to share some of our work and talk about what we've seen in the industry in the past, what we have seen in human biomedicine, and how that information can be translated into benefits or potential solutions in animal agriculture. So I apologize for my first slide actually adding a question mark to the end of that title. And really we need to identify the state of the art, which is we're just at the very beginning of identifying the mechanisms underlying transgenerational inheritance in animal agriculture. And we have more evidence in humans, primarily from epidemiological work. But I will give you some data today, some looking back and some looking forward, to tell you what I think the status is going to be and the potential for the future. So today I'm going to give you first a little overview. Scott gave you a brief overview of my research program, and I just want to show you how this work that we have undertaken fits into the general scheme. I'd like to talk about some observations that we made about 20 years ago now with the manipulation of calcium phosphorus levels in early poultry diets, because that was really the basis of where we've gone since that point. Some impact of long-term impacts of environment, including the diet, on phenotypes, and that's both within a generation and across generations. A little bit of a review on the status of epigenetics and the one-carbon metabolism mechanism, because we'll refer a lot to DNA methylation. My lab has focused on DNA methylation as a way to track epigenetic inheritance from one generation to the other, primarily because of the ease of its measurement. And then we'll talk about challenges in poultry production, and I'll give you some examples along the way, both in animal agriculture, in human bioscience, as well as in other species that you probably have encountered. And then the target being answering that last question, can we feed the next generation or next generations? So I will lead off with one of my frustrations about nutrition research from the perspective of a geneticist or molecular biologist. I was trained to want to know what the mechanisms are, and all too often I see my colleagues publish papers that describe, one diet being superior to the other diet. My first question is, well, what's the mechanism? Why is one better than the other one? And it's not always about body weight. I'm thinking about broiler production in poultry, but it should be about the entire package. So I'm also going to give you a little philosophical position that was ingrained in me by a mentor back at Virginia Tech, Paul Siegel. And as a true geneticist, he believed that there is no such thing as a free lunch. So no matter when you see advantages from whatever experimental manipulation you're engaging in, there's always a price to pay somewhere. And it's important to try and understand what that price is when you choose to alter a protocol or change a management practice. And to better sort of sum that up, I believe that there has to be a resource allocation model where the animal has potential. There are genetic resources available there, but ultimately the pie is finite. There is no 110%. There's 100%, and you reallocate resources between one category and another, and you can choose how you want to make those allocations. Okay, so that's sort of my philosophical background of where I come from, and my sort of desire to understand the why and the how, not just what a solution might be, but how does that solution work in a given situation. So I see my research program encompassing genetics, nutrition, and immunity, and how they overlap and affect and then hopefully improve performance in particular environments. We tend to use tools, And those tools are how nutrition impacts the genome, how that may also impact the microflora or the microbiome inhabiting the GI tract of the animal. As Scott said, we've used selected lines, genetic lines, to try and understand specific phenotypes. One of those is immune response. And all of those fit into an omics platform, looking at genomics, metabolomics, proteomics, transcriptomics, so all of these variables, and to just, or really tools, to give you an idea of some of our research areas. We are looking for regions of the genome responsible for quantitative traits. Those are important for phenotypes in animal production, but we've also done some work with microbiome. We've looked at feed ingredients that may have impacts on performance. And so we're using our toolbox, which are these molecular tools, RNA sequencing, genome sequencing, and some epigenetic sequencing, to try and understand what the mechanisms are underlying how traits are transmitted from one generation to the next, and within one generation as well. So I told you I was going to tell you a little bit of a history story. So how did we get into this arena or this area? So it started about 20 years ago, and when I was at USDA in Beltsville, I had a good friend and collaborator, Rosalina Angel, at the University of Maryland, and we started off trying to solve this problem. And so this problem, and this you'll see the slide is from the time period. This is a view of every county in the United States, and it's color-coded to describe the amount of manure phosphorus that's available to spread on the land. Okay, so this is where we came from. We wanted to figure out a way, could we minimize the phosphorus in manure? Therefore, we wanted to maximize the absorption of the animal so that we could formulate the diet and everything went into the animal, and ultimately we would love for nothing to come out the back of the animal. So our first experiments looked at early nutrient restriction. And so this is a very simple experiment where we limited, or we reduced, calcium phosphate levels in the diet. And so this was just a simple diet in that we had a control diet and a low level of calcium phosphorus. We treated that for 19 days, and then we put the birds, this is broilers, on a restricted diet of calcium and phosphorus. And what we did was we compared the outcomes out here at 32 days. And what we saw was that this conditioning treatment, this early restriction, resulted in improved efficiency of nutrient absorption out at the end of the trial, out at 32 days. And it's like, okay, what's the mechanism for this? Do we need to go 19 days? Can we shorten it? Can we create a program so that management could have this long-term influence on nutrient absorption? And so we played around and ended up with a model in where we reduced calcium and phosphorus for the first 90 hours. So that's a very short time period. And so in that first 90 hours, we reduced a dietary calcium and phosphorus to about 20% of the NRC level. Then we returned them to control diets, and we had paired treatments. You'll see those in the first and the second row of the treatment design. But what we did at 22 days was we shifted the birds back to a diet low in calcium and phosphorus for challenge. And what we found out is out here at day 38, when we measured performance, we had birds in this early conditioning period that had increased body weights, better feed conversion, increased calcium and phosphorus absorption, and increased bone mineralization. So this little time period here early had some long-term impact on how that bird performed throughout its lifespan. And so, being the molecular biologist working with the nutritionist, I went and looked at the phosphorus transporter in the GI tract. And what we saw was after that early 90-hour conditioning, we saw a dramatic upregulation in the expression of this phosphorus transporter. And let me see if you can see my pointer here. So in the low, we saw this an upregulation of about twofold. Then when we waited till 38 days and looked at those birds, there was also this marked increase in expression. So the brain, the wheel started turning in the brain, and we tried to figure out what could be the possible mechanism of this observation. And so we went to the human literature and found that there were some options, and there are multiple potential mechanisms. We didn't think that there was altered structure of the organ. We did some sectioning and didn't see that in their GI tract. There really wasn't any anatomical observations that we could see. And so we went and we knew that there was an effect on gene expression, so we started looking at regulatory mechanisms of the genome. And the thing that popped out of the literature that got us very excited was epidemiological studies in humans that were tracking human health from the 1940s all the way up through the 1980s. And this was led by a person in the UK named David Barker. And David found a relationship between humans, people, who were extremely calorie restricted during occupied Netherlands during World War II, and individuals born during that period had a much higher incidence of insulin resistance or diabetes. And the interesting thing was that not only did those individuals have that phenotype, but so did their offspring, even though they had a typical or normal diet. This concept or this hypothesis of the fetal origin of adult disease has been very thoroughly confirmed with epidemiological data, and can be linked to a number of health-related issues, including heart disease, high blood pressure, and as I said earlier, diabetes. And so this gave us some hope that maybe what we were seeing was a similar mechanism in our agricultural animal. When I was at USDA, we also had a honeybee lab, a lab that was working on pollinators, and it never really occurred to me until I started doing some literature and had coffee with one of the faculty there. And this is exactly what's happening during honeybee reproduction. So the queen and the worker bees are essentially identical genetically, except the queen is fed royal jelly. And this unique food source or nutrition source inactivates DNA methyltransferase 3. So genes that would normally be repressed are activated in the queen. And when it's active, those insects change morphology, change physiology, they increase their lifespan, and they have lots of different phenotypes from essentially their identical sisters. So this is within generation. So this is a long-term effect caused by nutrition early in life that is expressed through the lifetime of the animal. So we started looking for other examples of where it may have longer term effects. And the other example that we found was in locusts or grasshoppers. And so this is just a comparison of the two, where epigenetic effects, and I'll describe those on the next slide, in the example of the locust, are transmitted from one generation to the next, whereas in honeybees, it is developmentally programmed. So that epigenetic effect is within the single generation. So the grasshopper, or the locust, has two distinct phenotypes. There's the gregarious phenotype, which we equate to a locust infestation or destruction of crops, and the solitary form, which is a grasshopper. And those two versions, or those two phenotypes of the same individual are generated by a few stimulus. So high density, if you put lots of grasshoppers in a high density, the solitarius form, they will transform into the gregarious form within their generation. But if you take the gregarious locus out of that high density environment and make them solitary, it takes 2 to 3 to 4 generations for them to return to the solitary phenotype. And so that transformation persists from one generation to the next. And it's fascinating to me, biology and its complexity. So the mechanisms underlying both of these two observations in insects rely on modifications of DNA, not to the DNA sequence itself, but to other signals or tags that are layered on top of that DNA sequence. And these include, in most general terms, DNA methylation, where a methyl group is added to the cytosine residue of a double-stranded molecule, and acetylation of histones to which the chromatin is wound. And the degree of acetylation or methylation determines how active and open a segment of DNA is, and is it available to produce the genes that are encoded there. So it's an open and closed sort of mechanism. So we started searching for other examples of where dietary modification had direct connections to DNA methylation and then direct connections to phenotypes. And so in the literature, and just down the road at Duke University, Randy Jerdel's group was working on the agouti mouse. So this is a mouse that has a genomic insertion, and that insertion is in a region where methylation is important for regulation of the expression of the gene. And so what happens is, what you see here are identical mice, essentially, but their mothers were fed different levels of methyl donors in their diet. And so what happens is, if you increase the methylation potential within the in utero environment, the mouse will display a pseudo-agouti phenotype. So it loses the typical golden color, and it also is a normal body weight. It is not obese. So there is a mechanism that can control this. In mice, it resets during that methylation pattern formation resets during gamete production. And so it resets at the next generation. That's not always the case. I want to give you an idea of how important thinking about nutrition is from the human perspective, and then I'll give you an analogy in poultry. So I like this slide, and I believe it came from a UK campaign to reduce smoking. And so what you see here is, and I'm just going to read it because I think it's important to say what it says. A woman who smokes while pregnant induces epigenetic changes in 3 generations at once, in herself, in her unborn daughter, and in her unborn daughter's reproductive cells, because all of those, all of the eggs present in that unborn daughter are already there and being exposed to the in utero environment of their mother of generation 1. And the observation is that environmental exposure causes long-term health impacts. And you should be able to see from this cartoon is that our generation 3 is experiencing obesity and perhaps other metabolic diseases. So this isn't the only example. Smoking is not the only example. There are plenty of examples. I'm going to give you a brief overview, and if you would like to read more about all of these examples, I suggest you take a look at a reference, King and Skinner from last year in 2020. And so these are the species that are known to have transgenerational effects that are carried through epigenetic modification. Plants through humans, and you'll see birds are in there, and we'll talk a little bit more about birds in a minute. But you'll also see that there are environmental impacts that are beyond nutrition. So there are environmental hazards and toxins that have been shown to have epigenetic effects across the generations. But nutrition is one of the types of environmental exposures, as is stress and trauma. Exposing individuals to enhanced stress causes epigenetic changes that are transmitted across generations from one to the next. So there are examples of nutritional effects with specific nutrients. There are also examples of caloric restriction. There is quite a few well-described studies in rodents looking at caloric restriction and how that impacts general health and metabolic diseases multiple generations down the line. I just want to give you another analogy as to why we will be talking about human and comparing it to poultry as a specific model. There is a generally accepted idea in human nutrition and that early establishment of homeostasis in humans as they develop, that the first 1,000 days are critical, are really important. And that's the first 1,000 days from conception up through sort of the toddler age. And the interaction of maternal and neonatal and infant nutrition with the microbes inhabiting the GI tract of the individual, as well as the epigenome that they have inherited from their parents. So by analogy, that first 1,000 days of life, if you compare a chicken's lifespan to a human's lifespan, is really equivalent to about the first 63 days of life, which is 21 days inside the egg and another 42 days outside of the egg. In broilers, that's about the extent. So if you make those analogies, you could infer that a broiler is in its entire life before it goes to market within this similar window of opportunity to make changes. So I think it's a reasonable model to think about for human and other agricultural animals, as well as how the integration of nutrition, the microbiome, and epigenetic factors impact health. Okay, so we wanted to focus on birds because that's where we live on our campus, and we wanted to try and identify transgenerational evidence for inheritance of epigenetic marks and impact on things that we care about in animal agriculture, which are phenotypes that relate to animal production. So I'm going to talk about a study that was just finished by one of my PhD students, Chelsea Phillips. This was a study to evaluate dietary methyl donors' effects on reproduction and performance from one generation to the next. And our goal was to evaluate the effects on gene expression, phenotypes, first of all, and performance traits, and then on gene expression, and then on DNA methylation patterns to see if we can see this epigenetic effect of diet transfer from, one generation to the next. And we know there's been a recent interest in nutritional programming in livestock. However, this is going to be was done in a poultry species, particularly the quail, and we chose the Japanese quail because of its extremely short generation interval, because we could do things in one generation and watch for their effects downstream. So in this study, and what you will see is we had multiple generations, so there's 3 generations in the study. The only birds that were fed the diet that included increased methyl donors, and I'll describe what that diet looked like, was the first generation, so generation 0. and the only birds that received the diet were on this left half of the table here. And so once they were exposed to that high methyl diet, either in the starter or in the layer diets, then all the animals were returned to control diets, and those were control starter or control layer, and then produced through sexual maturity, eggs, incubated, hatched through the second generation and the third generation. And so we looked at the phenotypes. The enhanced methyl diet included about approximately 10-fold NRC level of choline chloride, betaine, and the vitamin B12, B6, and folic acid, as well as zinc sulfate. And I do have that on the next slide. And all of those components feed into this one carbon metabolism pathway. And these function to produce amino acids like methionine. That's a valuable component of a cell and valuable nutritionally, but they are also involved in the cycle that regulates DNA methylation. S-adenosylmethionine methionine is the source of methyl groups for DNA methylation. We also didn't want to manipulate the amino acid balance, so we did not feed additional methionine, and we didn't add riboflavin, which we might have been able to do, but solubility was an issue for us. So let me show you what those levels were in the methylation package. That's Dr. Phillips's terminology. And so we increased those components to these levels so that we had about 10-fold what would normally occur in the basal diet. We looked in our trial at various parameters. We looked in the generation 0, we looked at body weight, number of eggs produced, egg weights, body composition, and then we had a lot of molecular phenotypes that we measured in each one of the generations. So I'm just going to give you an overview of what we saw that was significantly different. And what we saw was in the G0 parents, we saw an increased body weight of the birds that received the methylation diet. We saw an increase in egg weight, and we saw an increase in liver weight. And we think this has a lot to do with that increased methyl donors. What was interesting was we saw a continuation of body weight influence, and this has got to be related to the size of the egg from G0. But what was interesting was in G1, the first generation, which had not seen the methyl diet, we saw an increase in egg weight as a function of treatment. That phenotype continued into the second generation. So generation 2, when we looked at and followed the treatment back, we didn't see any effect on body weight or any other variable that we measured, but we did see an increase in egg weight. I have that on a graph here to give you an idea. And so these is a description in generation 1, the treatments of their parents, the number of eggs measured for each one of the treatments, and we see that those birds that had high methyl in the starter phase had significantly higher, heavier eggs than the birds that were in the alternative formulas, either the low starter or control or the low for both treatments. We also saw an effect in the second generation on egg weights. What we saw was a little bit different because there was an inversion in the phenotype. In the second generation, we saw that the birds that were whose grandparents had the high methyl diet had significantly lower egg weights from the birds that were fed the control diet. In addition, in this second generation, we looked at the bird's response to an antigen. We use sheep red blood cells as our antigen. The birds were injected with a 0.25% solution. And then we followed 5 days later to see what kind of titer they would produce. And what we saw was birds that had high methyl donor in the starter diet of their grandparents had a higher antibody response than birds whose grandparents had not seen that high methyl starter diet. So we saw phenotypic changes across multiple generations. So then we started looking for changes in DNA methylation. And what we saw was through bisulfite sequencing or methylation sequencing, we were covering about 70% of the available sites in the genome where DNA methylation occurs. These are in CPG regions. These are regions that flank coding genes. And what we saw was that there were a very small percentage of the total methylation sites that had enhanced methylation or hypermethylation, and some that had reduced methylation or hypomethylation. So those were very small percentages of the total. We then could look and see what the genes were adjacent to those methylation differences in the genome. And surprisingly, one of those regions was directly connected to that phenotype that we measured of antibody production. So MHC class II molecules were altered in their methylation status, both hypo and hypermethylated regions. And so that gave us some evidence that our phenotype that we were seeing was connected to the methylation status of those individuals. So some summary and a little bit of insight. So we saw in an avian model that diet can impact offspring for at least 2 generations. We need to go back and try and look at those individual loci that are known in the chicken genome. We'll have to make an analogy to the quail, that are affecting egg size, and see if there are any differences in methylation patterns there. We are treating both males and females. We didn't do this in an individual sex. That might be interesting too. But the other question is, well, what about a disease challenge or some other stress that might shift the phenotype when there's an abundance of methyl donors available in the diet, and there might be the tendency to reformulate or to adapt the genome to a given environment. So that leads me to sort of my last bullet point, talking about current industry practice in broiler breeders. And if you're familiar with that industry, it's structured like a pyramid in which pedigree stocks are selected for traits of interest. Those pedigree stocks are intercrossed to produce great-grandparents and grandparents. Usually this is a 4-way cross when you get to that level. And then those birds are expanded and broiler breeders are produced. And so those individuals are the parents of the broilers. So they have to have satisfactory reproductive efficiency. And so what happens is, in practice, is these top layers of the pyramid are in general not dramatically restricted fed. When they get to the broiler breeder level of the amplification, these birds are significantly feed restricted. Depending on the source of the breed, this could be as much as 40% reduction in ad libitum feed consumption. But then we expect their offspring to be placed in barns and eat. We provide ad libitum feeding, and those birds appear very driven by feed intake. And for the industry, that's a good thing. I am really interested and curious as to what is happening in the broiler breeder level of amplification that may be influencing the feeding behavior of the broilers. And I really feel that this could be an epigenetic mechanism. So One of my other graduate students has partnered with a collaborator in Canada to evaluate the impact of precision-fed animals. So if you've not experienced this mechanism, will refer you to Martin Sweetoff at the University of Alberta, where they have developed this technology that very accurately feeds birds individually, monitors their weight multiple times a day, so that each bird can eat as many meals as it wants, so that it maintains a target body weight. And this can be adjusted from the target body weight of the producer, that might be Cobb or that might be Aviagen, or it can be adjusted up or down depending on how much larger or smaller you target your animal. What Martin has seen, and we've visited and collected some samples, is that breeders fed to increased body weights, their progeny have a better feed conversion ratio than birds that are fed traditional restricted fed or traditional fed with precision feeding to the target body weight of the supplier. So our interest is to look at what's happening in those birds at the molecular level. So we're trying to understand what's happening in the breeder generation as well as in the broiler generation. And we've already done some gene expression studies and found that there is altered stress-related pathways in both the breeding generation as well as the broiler generation. We don't know yet what the impact is on DNA methylation. Those samples still need to go off for sequencing. So I think this is going to be a mechanism to try and understand what's happening at the breeder level, where restricted fed is common practice, restricted feeding is common practice. How is that modulating or manipulating the epigenetic landscape of those animals? And then how is that translated into the phenotypes and the behavior of the broilers? I think that's would be a really interesting, it's a really interesting biological question to answer. So I'm going to sum up here with this slide, and I've really focused on poultry, in particular broilers, because of the rather dramatic difference in how the breeders are managed compared to their primary breeders and to the production birds. And I have highlighted a few areas on this slide or this diagram describing where I think intervention could influence what's happening in the production birds, in the broilers, that may be less cost in the breeder generation, or even less cost in the primary breeder generation, especially if you're adding in a component of the diet. If you're trying to feed 200,000 birds, that's much cheaper than feeding millions of birds. And so if there's an opportunity to manipulate the phenotypes of the offspring of the broilers by simply feeding their parents in a particular way or with a particular additive, I think that would have incredibly positive impacts on the industry and have a positive economic benefit across the bottom line. There are other opportunities. So stress is another example of where you could minimize stress to try and influence the performance of the offspring. There could be the influence of incubation. That's a whole different seminar that I'm sure I wouldn't be the first one to give the presentation, but there are quite a few others who could talk about that and how there's influence happening from one generation to the next just through the incubation mechanism. Okay, I'm going to stop and acknowledge my team that has helped work on all these projects throughout the years, and funding sources from USDA and the North Carolina Ag Foundation, and particularly my good longtime friend Rosalina at the University of Maryland. And I will stop here and ask if anyone has any questions.
Thank you, Chris. Before we get started answering questions, we'd like to share a brief video, and then we'll be right back to answer the questions submitted during today's presentation.
As the global leader in choline, Balchem has spent more than 50 years perfecting the art and science of choline chloride production. The new Puricol line delivers the highest standards of quality, produced in state-of-the-art manufacturing facilities, and backed by the strictest process controls for a level of purity, safety, and consistency you can't find anywhere else. Turn to Puricol choline chloride from Balchem for an unmatched level of quality you can trust. Visit balchemanh.com to learn more.
As a reminder, you can still submit questions through the questions pane in the attendees control panel. Dr. Ashwell, our first question is, are there examples of other transgenerational effects in chickens?
Thanks, Scott. So yes, there are. I'm not sure, am I sharing still, or can I share my screen? Oh, here we go. Let's see. So this slide describes a study that was done back in the, well, early 2000s, in which this is a group in Europe. We're trying to understand how stress affects learning. And so the stressor on these animals, or these birds, was random lighting programs. So the lights in the bird's housing facility would turn on or turn off at random. And if you know birds, you know that they are very photorefractive, and they like a specific period of light during the day. And so what they were looking at was the stressed birds took longer to figure out that they had to walk down a long corridor to find food. So that's the description of the study there in the center. So the birds were placed at one end of a long corridor. They had to learn where to find the food by walking down the long corridor and then making a left or a right turn. So the birds that were stressed took much longer to figure that out. They were delayed. So when reproducing the birds, they ran out of control birds to produce the next generation. And so the lab person that was in charge of the birds used stressed birds as well as control birds to produce their next generation of test animals, and just kept track of who was who. And what they found was the offspring of the birds that were stressed also had a delayed learning to discover the feed at the end of the long corridor, even when they were in standard lighting conditions. So that just demonstrated that effect, that stress effect, was being transferred from one generation to the next. I thought it was a great example.
Yeah, very interesting. Next question comes from Carrie. What do you think would be the best commercial application to utilize these epigenetic changes?
And I'm writing it down because that's the way I roll. Best commercial application. Well, I will tell you, Scott, and I'm not sure who the question came from. When we started working with nutritional conditioning and that early nutrient reduction, it's very difficult to talk someone into creating a deficiency in young birds when everyone wants to make sure the birds survive, and a dead bird is not a profitable bird. And so that type of program was difficult. Now, I think there are some examples of commercial operations that have adopted a similar idea and tried to convince animal production systems to use that early manipulation to influence the phenotypes later in life. I'm not so sure it was all that successful. What I think is where there's potential is by providing additional resources, as we did with the methyl donors during reproduction, so that the offspring have all of the resources they need to adapt to the environments that they're being produced in. And I think that will allow for the optimal set of the environment at its given set of conditions. I believe there are some good examples of this in humans where prenatal vitamins have been prescribed, and there ultimately is a sweet spot that if you have too little, there's negative effects. If you have too much, there's negative effects. So determining what that titrating the benefit would be something that would have to be done in animal agriculture. But that's my feeling, is that providing adequate resources for the methylation patterns and for that matter the acetylation of the histones to be established easily during development would provide advantages to the offspring.
All right, thank you. Next question comes from Jaquez. Have there been similar results seen in swine? If so, are there any of them, as you said, inverted?
All right, similar results in swine. That I don't know. I do know that there has been effects within the first generation. The population structure is a little bit different, and so when variation occurs in swine, it tends to be small. And when you are looking at offspring, so if you manipulate one individual in poultry, you can look for shifts in hundreds of offspring. Rather than in swine, you can look for shifts in offspring in the maybe 20 or 30, depending on the number of litters. And so I think it would take quite a large study to be able to see the differences across more than one generation. What I will say, so I appreciate the question. I think the jury is still out. The influence, there's extreme influence during pregnancy, during embryo development in utero, and I think that's where the biggest opportunities lie, so that you could manipulate sound nutrition before delivery and have a long-term impact on the offspring. I haven't seen an inversion though. I believe I'm just going to make an anecdote. I believe that could be related to what is seen in nature in wild birds when there are abundant resources. Birds will tend to favor or produce one sex offspring over another because of trying to spread their genetic material. Compared to when the resources are scarce, they will selectively produce more of the alternative gender.
Oh,
so when resources are scarce, they tend to produce more male offspring. And when resources are plentiful, they tend to produce more female offspring. And if you start switching those diets around, you can see that inversion happen.
Very interesting. Jerome would like to know, how important is dietary fats on generational performance?
So the effect of dietary fat, so that is going to influence, particularly in birds, it's going to influence the composition of the egg and the yolk, which are the energy source for the developing embryo. If we're talking about it in other monogastrics like swine, there's going to be an influence on partitioning. I've seen some studies that have included other lipids like conjugated linoleic acid that have effects on partitioning of lipids, and they affect body composition, and that effect can be translated from one generation to the next.
All right. Next question is, you mentioned that the epigenetic effects last 2 generations. Do you notice the same difference in both generations, or does the difference decrease each passing generation?
Right, so the difference in theory should decrease every generation because you're diluting out the signal, or you're diluting out the signal. I guess that's best analogy. And it depends also on the exposure of the alternative parent. So if both parents are exposed to the treatment, both are going to be contributing to the epigenetic profile of the offspring. Something that we haven't done, which I think would be interesting to do, would be to affect only one gender, treat only one gender, and then follow that with matings to control animals to see it. My theory would be that it would decrease at a more rapid rate because only one of the parents has been exposed to the treatment.
All right. Next question comes from June May. How do you view genetic selection for certain traits, comparing to nutrition and environmental impact? Which one is more profound? They're putting you to the test.
I know. Okay. So I have to go back to a study, and I didn't put a slide for this in. I have to go back to the study by Havenstein et al. Here at NC State, and I think it was 2001 or 2003, where they took broilers from 1957, random bred broilers that were maintained without selection for 40 years, and compared them to commercial broilers that were contemporary for that time. And they fed them 2 different diets. They fed them the contemporary diet, and then they fed them the diet that was formulated back in 1957. And that comparison of genetic line versus nutrition pointed to about 80 to 90% of the improvement in production is a result of genetic selection, and the other improvement, 10 to 20, maybe 25%, depends on which replicate you look at, is a result of nutrition and management improvements. So I have to say, in the long term, genetic selection far outweighs what can be impacted by altering diet or management. But I think the real world issue happens when feed ingredients, resources change, formulations have to change. This happened when the ethanol industry grew and corn was more expensive, and so there were alternate diets, alternate feedstuffs, and so those birds that were grown in those periods had to adapt to those alternative diets that they weren't selected on. And so I think that's an example where there has to be rapid evaluation and rapid turnaround and rapid adaptation by the birds, and they are very capable of doing that.
All right, thank you. Glenn would like to know, why do you think there was a G2 inversion in egg weight in the one study?
I really think, so thanks Glenn for the question. I really think that the influence inverted because of loss of methylation. And the control birds remained at the control level. And I really feel that the birds that whose grandparents received the high methyl donor diets and had higher egg weights in the first generation, the reset was occurring. And when the reset occurs, it actually overshot the mark. And so what was happening is they actually produced smaller than average eggs in that second generation. We've got a lot of work still to do to sort out the mechanism for that. We're lucky that there have been quite a few studies in chickens identifying regions of the chicken genome that are associated with egg size traits. And so the plan will be to go back and look for that similar or homologous region in the quail for which we have methylation data for. And if my hypothesis is correct, then there's going to be a flip in the methylation status from generation 0, same with generation 1, and then it will flip to the opposite status in generation 2. We'll see.
All right, thank you. And I see now we're at the top of the hour, so we've got time for one more question. Clay is asking, what about folate? Pregnant mothers are advised to supplement with folate. Could that have a negative effect?
Okay, so folate is interesting. Folate was recommended as a solution for neural tube defects in human development, and it solved it. It was I shouldn't call it a magic bullet, but it really was a solution to a real problem. What has occurred since then is, and this is very common with human supplements, is if 1 pill a day is good, then 5 pills a day must be better, and maybe 10 is even better than that. And so there's not very many studies, but there have been a few that have looked at folate and identified folate levels in pregnant mothers, humans, and then followed their children over time. And what has surfaced is a somewhat sticky connection to autism, and I find that really interesting. In that, there was some evidence that with mothers that had lower than average folate, there was an increased incidence of autism, and mothers who had increased much more than average folate for the study group, and I can't remember how many individuals there were. Those that had much higher folate also had a higher incidence of autism, like 2 or 2.5 times the incidence. So I think the magic is moderation, and that every person is a little bit different. They're not exactly the same. And so by saying one recommended level is the right one for you, I think is a little naive, and that there has to be some tuning. One lucky thing that we do have in animal agriculture is that our animals have a much more defined genetic background, and so that we can plan and tune much better, much more easily than we can in the largely outbred human population. Thanks for that question.
Yeah, thank you very much. And thank you everyone for attending today's webinar. If you have additional questions, please submit them to anh.marketing@balchem.com, and we'll forward them to you along with the unanswered questions from today's session. Our next webinar will be on June 8th, when Dr. Javier Razo from AB Vista will outline the impact of enzymes and fiber in gut health. You can also join us on June 1st for our next ruminant-focused webinar. Dr. Bob James with Down Home Heifer Solutions will discuss the most important steps in optimizing calf health. Balchem's podcast series continues to offer a deeper dive into our webinar topics. We now have 14 episodes out on YouTube, your favorite podcast platform, and at balchemanh.com/podcast. This podcast series goes behind the scenes to be a part of the conversations that take place over a few drinks with friends. Search for Real Science Exchange on your favorite podcast platform and be sure to subscribe. On behalf of Balchem and Dr. Ashwell, thank you for joining us today.